So my name is Jacques de Priya, and I am the Clinical Automations Product Manager at Whitehead Scientific. Today, I have the pleasure of introducing Omer Clement, who is the Director of Oncology Products at LGC SeraCare. Omer will be telling you more about the solutions for oncology, NGS assay validation in a talk entitled Addressing Challenges of NGS Assay Validation with Precision NGS-focused Seraseq Oncology Controls. Please feel free to reach out to us as proud distributor of LGC SeraCare products in South Africa if you do need additional information or pricing for these products. And without further ado, I will hand over to Omer to take the stage. I'll stop sharing my screen, Omer, and you can take over.
Thanks, Jack. Let me share my screen. Can everybody see my screen?
Yes, we can.
Yes, awesome. So I want to thank everybody, and I want to start by apologizing for my technical challenges this morning here in the United States on the East Coast. Thank you so much for having the patience to wait. I want to mention that I am joined today by two of my Seraseq LGC colleagues, Rami El-Kwazai, who is our International Distribution Manager, and Dr. Christina Nalik, who is our EMEA Field Application Scientist. I am from the LGC SeraCare. We are a multinational corporation with 47 sites in over almost 20 countries. We have also 19 manufacturing sites, and combined, we manufacture and sell over 230 IVD label products. I am within the Clinical Diagnostics business unit, which is part of our Standards division. And within Clinical Diagnostics, we have clinical genomics, molecular diagnostics and serology, clinical chemistry and immunoassays, and native antigen and virology. Today, I will be focused solely on the clinical diagnostics unit, which has been a supplier to the IVD industry for over 30 years. I mentioned that we have multiple locations within the clinical diagnostics. We have our clinical chemistry and immunoassay based out of Portland, Maine in the United States. Our molecular diagnostics and serology are based out of Milford, Massachusetts. And today I'm speaking to you from Gaithersburg, Maryland, where we have our clinical genomics and next generation sequencing R&D and manufacturing facility. In this facility, we focus around oncology and non-invasive prenatal testing products. We have our R&D team, our manufacturer and QC, as well as our shipping department. Here we make RUO and IVD labeled products. So just looking at the capabilities we offer within the SeraCare Clinical Genomics unit, we have a range of portfolio products from infectious disease, NGS-based controls, inherited disease controls. In oncology, we have tumor profiling and immuno-oncology, which I will go into that in a little bit more detail. And we also have a circulating tumor DNA, which supports both our oncology portfolio as well as our non-invasive prenatal testing products. So we have all of these capabilities. They are designed and built specifically for next generation sequencing assays. So when we think about reference materials and how it impacts all of the different workflow stages within next generation sequencing technologies and products, we use and we believe that reference materials are available and applicable to assess technology and to optimize assays early. Also used, of course, in assay validation and QC for both a positive sample control as well as a workflow assay control. And reference materials play an important role in laboratories that wants to develop their own tests and submit this to local or regional regulatory agencies, or as well as federal regulatory agencies, whether this is CE mark, FDA, US FDA marked, or in other local regional locations. And finally, reference materials are critical to clinical lab operations in terms of guiding the ability to QC and reproduce data with high quality precision and testing, so that it can always provide high quality results to clinicians and patients that need the results coming from this clinical lab test. So reference materials are extremely important across all of these different workflows. Specifically to next generation sequencing, I show here just a generic workflow of NGS, starting from extraction of patient samples, through library prep, to the actual sequencing of these materials, to the analytics, and then the report generated to be sent back to the hospital. Within this entire NGS workflow, various parts where the products we build support our Seraseq Oncology products. And in sample extraction, we build encapsulated as well as formalin fixed and paraffin embedded reference controls that support the ability to have effective QC of both the quality of the extracted nucleic acid as well as the quantity and amplifiability of these nucleic acid materials. For library prep, we have pre-purified nucleic acids as genomic DNA, purified RNA, or fragmented circulating tumor DNA materials that can easily go straight into the library preparation stage of the NGS. And you can just compare that to the actual sequencing samples for the patients. Overall, these reference materials provide positive reference controls to make sure that the variants being called pathogenic or VUSs are matched to known variants in the control, giving confidence to the quality of the assay and the results being generated. So I mentioned all of these controls are developed specifically for NGS. They are based on a well-characterized genome in a bottle, genomic background GM24385. It's manufactured in ISO 13485 facility. We are US FDA audited, and as I mentioned earlier, we're a global IVD manufacturer. To look at how we actually build our reference materials for our oncology, we began our operations about 7 years ago now. And we use a well-tested technology that requires biosynthetic DNA plasmids that are manufactured and then spiked into a highly characterized genomic DNA cell line background. And this approach allows us to design patient-like performance with these biosynthetics. And we can build this as a mutation mix, purified variant, multiplexed variant content. And it can be used directly, as I mentioned, either as a sample control from extraction or as direct into a library prep stage of NGS. Because these are biosynthetics, we design them in a highly multiplexed fashion. We can design in any variant content, whether these are SNVs, indels, copy number variants, as well as structural variants. especially variants that are very challenging and very, very rare. And I show here many of the publications that underpin the technology for these biosynthetic reference materials. In a cartoon representation, just kind of give a quick overview. So we designed these variants at around 800 base pair size. The variants are spiked in the middle of this sized fragments. And these are mostly DNA plasmids. We can design in SNVs, indels. For our CNVs, they are full-length genes. And this combination of these plasmids are pulled together with a wild-type genomic DNA from Genome in a Bottle, cell line GM24385. So we pull these variants and the wild-type background together. We then quant that by digital PCR. The wild type is used to blend to the expected target allele frequency with the background, and we can formulate the mixture or bulk either as a purified mix in buffer or again as an FFPE or plasma. And then we do final digital PCR testing. We also include NGS because these products are meant for NGS, so we provide orthogonal NGS data to support the detection of these variants as well as the target allele frequency. And for the liquid biopsy controls, we do a couple extra steps. So from the pooled variants and the wild type background, we fragment and size this DNA to between 160 to 180 base pair to mimic circulating cell-free DNA in plasma. And again, once we've fragmented and sized to the required fragment lengths, we can then take this formulation as both a mutation mix in buffer, purified DNA, or encapsulated in a plasma mimeric for prior extraction to mimic plasma patient samples. So I mentioned we have our Seraseq Oncology products in two primary formats: as a purified nucleic acid or as encapsulated in plasma or FFPE preserved tissue reference materials. In some of the fill volumes and amount of nucleic acid we put per vial in a kit, they can range from up to 5 to 10x usage per vial, and that gives clinical labs good materials for replicate runs as well as provide them good cost model because they can reuse the same tube multiple times. So we've used this background to build out a range of Seraseq Oncology, Somatic Oncology controls. Primarily, we build them across 5 portfolios: immuno-oncology, solid tumor DNA, solid tumor RNA, liquid biopsy, as well as heme malignancy products. So while these are the individual products within these portfolios, we like to look at them as how they support clinical oncology applications. So when we look at clinical oncology applications, we look at them in areas of disease profiling and diagnosis, disease or treatment monitoring, treatment response specific to immuno-oncology-based therapies, as well as ability to build NGS assays that could go through a regulated control environment. So today, I will focus on how this range of Seraseq Oncology controls align with all of the clinical oncology applications used that supports NGS technologies. So the products I highlight in red here are products we plan to release next year. So look out for this. Let's start with solid tumors. So our solid tumor products will cover our DNA and RNA-based pan-cancer disease profiling and disease diagnosis. For solid tumor DNA products, we have a range of purified tumor DNA mutation mixes, as well as FFPE tumor DNA materials, and as well as copy number variant mixes. For our tumor DNA purified mutation mixes, we have 28 gene, 40 variant mixes. And these primarily have SNVs, indels, as well as structural variants. For the tumor DNA, we have all the variants plexed at either 7% AF or at 7 or at 10% AF. Of course, they are PCR and NGS quantitated. We have a unique industry first protocol, our tri-level DNA. 28 genes, 40 variant in a single reference material. Again, SNVs, indels, and structural variants in a single reference material. These have 13 variants at AF 4%, 13 others at 7%, and 14 at 10% in the same reference material, available again as a purified DNA. We released tumor FFPE DNA last year. We believe it's industry first because it allows ability to genotype all variant types in a single reference material. 17 genes, 34 variants, including SNVs, indels, copy number variants, as well as structural variants. Also built to be patient-like with a DNA integrity number range between 3.9 to 4.3. Standard or typical tissue FFPE from patients, about 3 to 3.5 in DINs. And this product was quantified using digital PCR and TSO 500. We have a couple CMV products, lung and brain cancer and breast cancer. They are amplified at +3, +6, +12. Each has 3 clinically utility genes known to have important copy number variant to those specific diseases. And so when I mentioned that we quant by digital PCR, here is for our tumor DNA. We have AF4, AF7, and AF10 by digital PCR across all 40 variants. For our fusion RNA products, we have several, and we have a pan-cancer fusion RNA product. We call it fusion RNA V4. We have 18 pan-cancer fusion genes, about 1,500 copies per microliter for each fusion gene, quantitated by digital PCR and NGS. And this product is designed to support targeted amplification-based and hype capture-based NGS assays. We have a whole transcriptome RNA-seq control, also an industry first. It has 22 pan-cancer fusion genes. Each fusion gene is plexed to about over 20,000 copies per microliter, quantitated by digital PCR and whole exome RNA-seq assay. As I mentioned, it's a control designed to support whole transcriptome RNA-seq NGS assays. We also built an industry-first NTRK fusion RNA product, both as a mix as well as FFPE. This was designed to support lab-developed tests as well as companion diagnostic assays that are used to validate anti-trypomyosin drugs that have NTRK fusion in cancer patients. So looking at our, for example, looking at our Seraseq fusion RNA mix product, we have multi-lab testing. 6 laboratories took these materials and ran this through their own individual assays. These assays included Illumina's TST170 and a range of Archer fusion plex RNA assays. Essentially, all 6 labs reported high sequence efficiencies, very high on-target map, detecting all 18 fusions and found to be compatible across multiple NGS assays, both hype capture for TST170 and amplification based for Archer FusionPlex, and RNA performance as low as 40 nanogram input. For the fusion RNA FFPE V4, we also did multi-lab testing. And here we talked to about we worked with 5 individual laboratories, and they used different extraction kits. Excuse me. And for the different FFP extraction kits, they used QIAGEN or PrEP, Promega Maxwell, as well as Agincourt former pure FFP RNA. You can see some of the extraction efficiencies depending on different RNA kits. Again, expected yields by kit. Maxwell kit was found to provide the lowest yield, Agincourt kit the highest yield, but more importantly, all 18 fusions were detected across 3 different assay types, a range of Archer FusionPlex, both custom as well as off-the-shelf, Oncomine focused assay, as well as Illumina's TST170 hype capture. One of the interesting results was published last year by the Fernandez Group at Columbia University was about how they used the Seraseq fusion RNA control to as a reflex test to their NGS DNA mutation assay. So the Columbia University Medical Center is a New York State Department of Health certified CLIA lab. They have a 47 gene assay for lung cancer. And this 47 gene DNA assay is clinically validated and approved by New York State. However, in some cases, some patient tests are missed because there are no DNA mutations, so they needed to reflex to a fusion RNA panel. To validate the fusion RNA panel, they used the Seraseq fusion RNA control, and they were able to showcase that reflexing to this fusion RNA panel improved clinical yield for additional patients that turns out to have important clinical markers for lung cancer, reducing the number of overall tests between 30 to 35% because of this reflex RNA panel, overall reducing the cost burden for the hospital. So this is how they use a product to evaluate and ultimately support their clinical laboratory as well. I'm going to move to the next one. Liquid biopsy products. We have a range of liquid biopsy ctDNA products for disease diagnosis and disease monitoring, and I'm going to walk through some of these products with you. So we have a ctDNA version 2. We have a mutation mix and a reference material. Has 28 genes, 40 variants, all of clinical utility in pan-cancer diseases. Covers SNVs, indels, and structural variants. Variant allele frequencies range from 0.125, 0.25, 0.51%, 2%. And we also have a wild type control as well. These products are qualitated using digital PCR and NGS. We released ctDNA Complete a couple years ago. This product is designed primarily for solid tumor diseases, unlike our V2, which is pan-cancer with 28 genes. Our ctDNA Complete has 16 genes and 25 variants, all with clinical utility in solid tumor diseases. And these are mostly SNVs, indels, copy number variants, as well as structural variants. The variant allele frequencies range from 0.1 to 0.5, 1%, 2.5, 5%, as well as a wild type. Again, quantitated by digital PCR and NGS. This year, we released an industry first ctDNA MRD panel. It has over 600 variants. It is built from a tumor cell line, and it's matched, SNP matched normal, as well as additional biosynthetic plasmid DNA variants, which includes SNVs, indels, as well as copy number variants. We have blended this MRD panel to tumor fractions at 0% as the wild type negative control, and then 0.5%, 0.05%, and 0.005% using both digital PCR and NGS. And I'm going to talk about this in a little bit. So comparing our ctDNA version 2 and complete, again, version 2 is a pan-cancer product, whereas our ctDNA complete is designed primarily for solid tumor products. And essentially, the complete was designed to allow ease transition from solid tissue FFPE based, which typically have LOD around 5% for the variants, and so we blended the complete to also up to 5% at ctDNA level, easing the transition between solid tissue materials to liquid biopsy applications. And the key difference, of course, also is that the ctDNA complete now includes copy number variants as markers within the control. So one of the interesting multi-laboratory evaluation that was coordinated by the National Institute of Standards and Technology here in the US featured NIST, National Cancer Institute, Boston University, Johns Hopkins, University of North Carolina School of Medicine, Gothenburg University in Sweden, as well as SeraCare, where they looked at everything from extraction efficiencies from plasma mimetics at various blended AF for the 40 variants in the ctDNA reference material. They looked at that efficiency on the left side, and on the right, they also typed for all of the blended variants at the allelic frequencies at 0%, 0.1 to 5, all the way to 2%. And you can see how all of the different laboratories were able to quantitate and identify all of the variants at the blended target allelic frequencies in the single reference materials. Another validation was performed by New York Presbyterian Hospital in conjunction with Weill Cornell Medical College. They had a clinical Oncomine cfDNA lung panel that they submitted through New York State Department of Health for clear. And what it looked at was look at variable allelic frequencies from our Seraseq ctDNA control. They found 11 variants that are concordant between our control and the Oncomine cfDNA lung panel. Using variable DNA inputs and sequencing coverage depth, they were able to hone in on an LOD of 20 nanogram input and a detection efficiency of about 0.5 allelic frequency using the Seraseq ctDNA control. This analytical validation data was included in the CLIA submissions to the for CLIA approval for this assay in the hospital. I mentioned we just recently released our MRD, but one additional validation performed by Sloan Kettering in New York has now been approved by New York State since 2019. The validation study and the clinical submission was published just recently in 2021. Here they have the MSK access. It's a cfDNA pan-cancer liquid biopsy assay, 129 gene panel, validated to detect all classes of somatic genetic alterations in cfDNA specimens. And part of the validation include using the Seraseq ctDNA complete at 1% and 2.5% tested in triplicate, And they also looked at all of the different blends at 5%, 2.5, 1%, 0.5, and 0.1. Ultimately, they were able to confirm the LOD for de novo detection of variants at 0.5% with 92% sensitivity, and overall 99% for known mutations in a patient sample. So this is a very, very exciting study published with over 50 authors or co-authors published this year. This assay is clinically deployed by Sloan Kettering in New York. So we recently released MRD product this year. I mentioned earlier that this product is a combination of tumor cell line with a SNP-matched normal. We also spiked in DNA variants from our ctDNA complete. This was blended at 0.5, 0.05, 0.005. A 0% blend is a wild type to identify any somatic variants present in the wild type that could be qualified when you look at the much more lower, higher sensitive variants. And the idea is actually similar to Natera Signatera MRD assay. So the idea here is to do an exome sequencing of patients, so you can have almost an exome level understanding of all of the somatic variants important to those patients, and select some of the highly pathogenic variants into a targeted panel, and then from plasma of the patients taken through the blood samples, blood draws, extract cell-free DNA, and then track those 16 variants across each of those patient population. This tracking of the ctDNA, positive or negative in the targeted smaller panel, was validated using our Seraseq ctDNA V2 product. And they were able to take allelic frequency 0.5 control, titrate it down, do a titration curve down to 0.005 to give them a good understanding of how they can easily track ctDNA presence or absence in a patient sample as low as 0.05%. So I want to quickly move to our immuno-oncology portfolio. By design, these are biomarkers for therapeutic response. In this case, tumor mutational burden, as well as blood TMB and microsatellite instability. To build these products, we had a global collaboration with a range of industry partners. We supported the Friends of Cancer Research TMB Harmonization Project with Genomic DNA TMB. In Europe, the IQM Path Proficiency Pilot Project with FFPE TMB, and with our SeraCare TMB Working Group, we developed both the gDNA, the FFPE, and the blood tumor blood TMB products. These reference materials are blended differently. So our gDNA TMB is 100% tumor normal paired. Our FFPE TMB is 30% tumor blends, and our blood TMB are blended at 0%, 0.5, and 2% tumor contents. Again, all of our products, all of our TMB products are derived from patient samples. We also have and develop our own TMB scoring pipeline and run this primarily by whole exomes. For blood TMB, we use targeted NGS panel. So our development is to take tumor normal matched tumor normal matched samples, expand, grow, and through different cell cycles, extract genomic DNA from the tumor cell line as well as the normal cell line, run this through whole exome sequencing. And for our FFPE, we pre-blended the tumor down to 30% tumor content, and then paraffin and embed and formalin fixed that. A typical WES data for tumor cell line, you can see the result at the bottom of this page. Mean exome coverage is about 200x for the tumor cell line, for normal about 100x. And at the top right-hand corner, I showcase the different TMB scores that we generated for 5 TMB materials, TMB score 7, 9, 13, 20, and 26. So we have both the genomic DNA TMB as well as the FFPE TMB scores. Moving quickly, these products were provided to the Friends of Cancer Research Consortia, 16 laboratories, and this was presented in 2019 at ASCO. Also, all of these products were built and evaluated across multiple targeted panels by a range of laboratories. And you see the dotted lines are the West TMB scores, and the the vertical lines are the different assays across all of the different laboratories. The objective here is to see how targeted panels TMB's measurements match whole exome measurement. We recently released our blood TMB products, and these products are derived primarily from the same tissue TMB materials, except that in this case, we are fragmenting and sizing and then blending these to 0%, 0.5, and 2% tumor fractions. In this case, we are quantitating this blood TMB solely by a targeted NGS panel. And that targeted NGS panel was the TSO 500 running on a NovaSeq 6000 with the S2 chip, analyzed through TMB pipeline, Dragon TSO 500 analysis software, input of about 30 nanograms run in replicates. And for score 7, 13, 20, and 26, you can see the adjusted TMB scores are fairly decent and close to the tissue TMB scores. Again, this is an industry-first product available only through LGC SeraCare. So here's a total SeraCare Seraseq TMB portfolio for TMB scores 7, 9, 13, 20, and 26, both as genomic DNA, FFPE, as well as blood TMB. We have a range of scientific collaterals to support customers who buy these products. We have technical reports. We have VCF files across every single tumor fraction. We have data sheets that collates all of the data generated by different laboratories, comparing to West or targeted panels. And we have also a number of papers and posters either already presented or coming out fairly soon. And finally, our heme malignancy products. These are blood cancer disease profiling products for leukemia and lymphoma cancers. Here we have for myeloid AML and MDS, we have DNA-based myeloid controls, 22 genes panel. It has SNVs and indels. It's available as a purified DNA. All the variants are blended to between 5 to 15% and quantitated using digital PCR and NGS assays. For myeloid fusion control, we have 9 myeloid fusion genes blended to over 1,000 copies per microliter for each fusion gene. Again, it's purified RNA format, quantitated by digital PCR and NGS. This year, we released industry first lymphoma panel control. It has 26 lymphoid cancer genes for both has SNVs, indels, and structural variants for Hodgkin's lymphoma as well as non-Hodgkin lymphoma biomarkers. All the 26 variants are blended to between 5 to 15% AFs and quantitated using digital PCR and NGS. Here is the validation by Thermo Fisher R&D looking at Oncomine, using Oncomine myeloid, the Seraseq myeloid DNA. You can see all 15 variants type fairly close between the target AF and the actual measured AF using the S5 platform. I want to finish by mentioning that we also provide custom product design with our VariantFlex, broad selection of variants we've already designed and tested and in our variant library, more than 400 variants across more than 60 genes. You can select from this, or you can provide us your own gene list, and we can custom build this for you. We can build from AF levels from 0.1 to 50%, or as many copies of RNA fusions you want. We are able to provide it in any format: DNA, RNA, circulating tumor DNA, any size of these nucleic acids, and any matrix, whether you want it in purified TE buffer, encapsulated in plasma, or FFPE preserved. And we provide all this information to you. You review and finalize the specs. We provide a quote, place an order, and we typically ship this within 8 to 12 weeks from design to delivery. To finish, I want to mention that we collaborate and partner across the globe with a range of stakeholders. They support us and we support them. We are members of multiple consortia, making sure that we are an important support vendor across all of clinical oncology and clinical genomics. And finally, just to summarize, our Seraseq Oncology product differentiators, they are NGS focused, they are highly multiplexed, they are very patient-like. They provide all genomic aberrations in a single reference material, SNVs, indels, CNVs, as well as structural variants in the same single reference material, allows you to standardize your clinical NGS assays. We provide both positive and negative controls, and we have a range of innovative first-to-market products, TMB, or blood TMB, whole transcriptome, ctDNA, MRD, lymphoma FFPE, products are all first to market. And with that, I will stop and take any of your questions. Thank you.
Thank you very much for a very informative presentation, Omo. We are going to open the floor for questions. If you have any questions, please feel free to raise your hand. My colleague Lauren will then unmute you, and then you are free to ask your question to Omo. Liesel, I think we're going to start with you. You did type a question in the chat, but Lauren, if you wouldn't mind just unmuting Liesel, just to give a bit more context on the question that she has, please.
Liesel, you are unmuted, and you're welcome to go ahead and ask your question now.
Can you hear me now?
Yes, we can.
Yeah, so my question was around ctDNA and how that is actually fragmented to the correct size. Is it fragmented via enzymatic fragmentation or mechanical sharing?
Yeah, it's a good question. So we typically don't provide our sharing method. I think we can share mechanically, we can share sonically, we can also share enzymatically. And I think we've tried all three. At this point, we find that enzymatic sharing with certain cleanup steps provides the cleanest circulating tumor DNA fragment sizes, allows us to also not just to fragment, but to also use a way to size to the specific ctDNA fragment lengths. So we do both fragmentation across, as I mentioned, all these three different types of approaches, but also use a sizing technology to size this to the right CT fragment lengths.
Okay, thanks very much.
You're welcome. Does that answer your question?
Yes, it does.
Good.
Thank you, Liesel. Is there anybody else that has a question to Omo? Omo, it seems that you were pretty clear in your message. So from Whitehead Scientific, we'd like to thank you once again for taking the time to present the solutions that you have on the NGS controls of LGC SeraCare. And thank you everybody in the audience for joining us. As I mentioned, please feel free to reach out to myself, Justin or Lauren, if you have need any additional information, or if you need any pricing on these wonderful products. Thanks everybody. Enjoy the rest of your day.
Thank you everybody. Bye.